Today we will be starting our third laboratory. We will be doing gram staining of 4 different bacterial colonies which we have isolated in last experiment. First we will observe last experiment's results. We did spreading of our mixed culture which has been grown from old stale water. In this first image we have added 10 microliters of sample for spreading. That's why we have got a lot of bacterium. I respreaded just 2 microliters of sample, and in this image you are able to see the individual colonies. This is how the spreading should work. I started with picking individual one type of bacterial colonies from 3 plates as shown by arrows and numbering. This video explains the procedure. That is, first we sterilize the dough in flame, then we leave it for 2 to 3 seconds to cool it down, and then touch on the agar to check how heated it is. After which we pick up individual colonies and streak it on a new plate. This was done for 4 different colonies. In these images you can observe 4 different plates with 4 different bacterial colonies having different shapes, surface and color. Then we start the next day. These are the reagents we will be using: crystal violet, Brahms iodine, decolorizing agent that is 95% ethanol, and counter stain as safranin. First we will number the slides according to our sample and draw a wax circle on the slide to mark area for the bacteria. Then we will add a drop of water for adding bacterium using inoculation loop. Next we will pick up a small amount of bacterium from the streak plate we had prepared yesterday on a loop after sterilizing it and add it to a water droplet. And mix it. Then we heat fix the bacterial cells by passing it over the flame multiple times till the water dries. After which we add crystal violet true drops to the heat fixed bacterium and leave it for 2 minutes. Wash the crystal violet with distilled water till excess stain washes away. Then add 2 drops of gram iodine to the slide and leave it for 30 seconds. After which wash it with distilled water till excess stain washes off. Next step is decolorization step, in which 95% ethanol is used as a decolorizing agent. Drip it over the stained part of the slide slowly so that excess stain which has not been fixed washes off. It should be done slowly. Remove the remaining amount of ethanol on the slide with tissue paper after this step. Add 2 drops of saffronin after the decolorization step. Add the heat fixed bacterium and leave it for a minute or so. After which, wash it with water in as in other steps. After all these steps, the slides are observed under a microscope and pictures are attached herein. First image is of Coccobacillus gram-negative bacterium. Second image is also Coccus gram-negative. Third is Bacillus gram-positive. Fourth looks like gram-positive, but as due to overheat drying, the bacteria is dead and its structure is distorted. So we cannot suggest with surety that it is gram-positive Bacillus.